So, hi once again, welcome to today's session. Today we'll be doing the development of the ear, and the scope for the session will be ideally to develop the ear in terms of its embryology. And before that, uh, is that you're supposed to know the anatomy of the ear, the normal gross anatomy of the ear. And the ear at this point is divided into three parts. It has an outer ear, a middle ear, and an inner ear. So starting us with an outer ear is that you have this auricle. Then after the auricle, you'll have this external auditory canal. And then after the external auditory canal, you'll have this tympanic membrane separating the outer ear or the external ear from the middle ear. Then the middle ear at this point will be containing the ossicles and their muscles. And then for the inner ear, it will be containing now the semicircular canals, the cochlea and the cochlear duct, the vestibule and the cochlear nerve, and then we also contain the vestibule and now the structures that are related to the vestibule. For example, we'll be containing the semicircular canals, the cochlea, and then at this point we'll also be having now the eustachian tube, which is also known as the pharyngotympanic tube. So the reason why it's called pharyngotympanic tube is because it connects the pharynx, pharyngo, and then the tympanic cavity. Um, that's the reason why it's called pharyngotympanic tube. Yeah, so those are the three parts of the ear: outer ear, middle ear, and inner ear. And then now for the auricle, it is divided into various parts, as we'll see in a few minutes. So it has this lobule, and then you have the triggers at this point. Opposite to it, you have anti-triggers, and then you have the ear notch. This is the anti-helix. And now, given this is a helix, this is the anti-helix, and then this is the anti-helical fold. You can see the upper cruise of the helix is a scapha, and then the lower cruise of the anti-helix. So you can appreciate this anti-helix, this is a cruise of the anti-helix, and then the root of the helix. So this is a helix, the whole of this is a helix, and then the whole of this is the anti-helix. There's an anti-tragus, there's a tragus, and there is a concha, there's a scapha. Ideally, you're supposed to know the anatomy of the auricle. Not forget that you have the navicular fossa at this point. The other thing that you're supposed to appreciate is that you're going to start with the development of the ear, and you're supposed to appreciate that the ear develops in a way that you have the surface external ectoderm occurring at the level of rhombencephalon. So remember, for the various brain vesicles, you have prosencephalon, then you have mesencephalon, and then you have the rhombencephalon. So this rhombencephalon is a hybrid at this point. So you have the surface ectodermal thickening occurring at the level of rhombencephalon. And then those thickening at this point, they will give us the otic placode. And then this otic placode, we can see them here. This is the otic placode. You can appreciate this is a developing neurotube. So it is closing at this point. This is a folding happening. And this is a surface, this is a neuroectoderm. So at the level of rhombencephalon, now we'll have the surface ectoderm undergoing thickening to give us otic placodes. And then these otic placodes, so the ear is also known as the otic. So the otic placode will invaginate to give us the otic vesicles, as we'll see now in this slide. So we have the otic placode, it invaginates to give us the otic vesicle. And then this otic vesicle is also known as the otosis or the auditory vesicles. So this otic vesicle now will get invaginated, will get into this mesenchyme that is inside it, and then it will differentiate into various cells, as we'll see in a few minutes. So the cells of the otosis differentiate into statoacoustic ganglion and vestibulocochlear ganglion, which is also known as the vestibulocochlear ganglion. So this otic vesicle coming from the otic placode, it will form the statoacoustic ganglion or the vestibular cochlear ganglion, and you'll see it in a few minutes on how now it will differentiate to give us the cochlear ganglion and also the spiral ganglion in a few minutes. So the otic vesicle divides into two. This is the the otic vesicle, which is now invaginating into two. It will invaginate, then it will divide into two, of which now the ventral part. So this is the ventral part. The ventral part will form the circle and the cochlear duct, and then the dorsal part will give us the utricle and the semicircular canals and the endolymphatic ducts. So remember at this point, we have the autocyst. This autocyst is this invagination at this point, which has invaginated and gotten into the mesenchyme. So some cells on the inner side will differentiate from this autocyst and form these ganglions. But the remains of the otic vesicles are the ones that will differentiate, of which now the ventral part will form the circular and the cochlear ducts. And then the posterior points now, or the dorsal part will give us the utricle, the semicircular canals, and the endolymphatic duct. So in short, everything that is being formed here becomes the membranous labyrinth, except the cochlear duct. So the cochlear duct doesn't come from these things that you have discussed here. So I hope that tries to give us a summary on how these things will come across. And actually, this is what we are talking about. So this is the autocyst. So the autocyst will differentiate into two. The ventral part is this one, giving us the circular and the cochlear duct. And then the dorsal part will give us the utricles and the circular canals and the endolymphatic duct. And actually, this is the endolymphatic duct at this point. The endolymphatic duct. These are the now semicircular canals and the utricle, then the circle and the cochlear duct will be coming from this. So you can actually appreciate at this point you have a developing tympanic cavity and the ossicles, but we'll come into that in a few minutes. So then how do you develop the remaining part, the circle and the other bit? So on the 6th week, the circle forms the tubular outpocketing at the lower pole. And actually that can be manifested here. So remember this is a circle. This is endolymphatic duct. This is a utricle. So this circle will develop some outpocketing. It will develop some outpocketing as you can see here. So this is outpocketing on the lower pole. So this is the lower pole. You can see the outpocketing is coming across tubular outgrowth of the circle. So this outpocketing will undergo spiral function. So the tubular outgrowth, this is a tubular outgrowth, which is the cochlear duct, will penetrate the mesenchyme. Remember, it was inside, the autocyst was in the mesenchyme. So it will penetrate the mesenchyme in a spiral function. And that's the reason why the cochlear duct is always in a spiral function. So this outpocketing, all the circle, will undergo outgrowth, which is tubular. And then this tubular outgrowth will penetrate the mesenchyme in a spiral function. This is the one that you're talking about. What will happen after that is that in the 7th week, you have the cells of the cochlear duct. So you have, this is a cochlear duct. The cells will differentiate from into the spiral organ of Corti. But before then, this spiral organ of Corti will undergo some formations. And this spiral organ of Corti is all that's the work of transduction of sound vibrations into electrical signals. So we've come to the point where we have the circle. This circle want to form the, remember this, we have a point in the inner ear that is known as the circle. The circle is the one that also gives the development of the cochlear duct. So what happens is that on this lower pole of the circle, we'll have a tubular outpocketing. This tubular outpocketing grows in a tubular fashion. And then after that, it penetrates the mesenchyme in the spiral function. In the 7th week now, the cells of this cochlear duct will differentiate to form this spiral organ of Corti. And then histologically that this spiral organ of Corti is the one that enables the transduction of sound vibrations into electrical signals. And maybe to finalize is that the cochlear duct, this cochlear duct remains attached to the circle via these ductus reunions. This is a narrow, the narrowing that is present in this cochlear duct and the circle. So the other bit we need to talk about is the development of the spiral organ of Corti at this point is that the mesenchyme surrounding the cochlear duct will differentiate into a cartilage. So this is a mesenchyme that you're talking about. This is a cochlear duct. This is now the future spiral organ of Corti. So the mesenchyme surrounding it will differentiate into a cartilage, and this cartilage is actually the one that will give us the bony labyrinth surrounding now the cochlear duct. In the 10th week, the cartilage undergoes vacuolization and hence two perilymphatic spaces, the scala vestibular and the scala tympani. So remember, histologically, we know that the cochlear duct is divided into three portions. We have the scala vestibuli, you have the cochlear duct, and then you have the scala tympani. So at this point, you can approach it as a cartilage, developing cartilage. But after this cartilage, you have a bone, the bony labyrinth. How does the bony labyrinth come across? It comes across due to the cartilage that is formed on the outer part. But now the cartilage that is inner part undergoes vacuolization. So you have this cartilaginous shell undergoes vacuolization. When we talk about vacuolization, you're creating space. So you create a space here on the upper side and then a space on the lower side. The space on the upper side forms the scala vestibuli, and the space on the lower part forms the scala tympani, separated by this is a vestibular membrane, then this is a basilar membrane, as you can appreciate here. So these two perilymphatic spaces, the scala vestibuli and the scala tympani, are formed, which are separated by vestibular and basilar membranes, as I had alluded earlier. So we can see three chambers at this point: scala vestibuli, you can see cochlear duct, and then the scala tympani. And now this cochlear duct, the cells of it will differentiate to form the spiral organ of Corti. And you'll see the formation of this spiral organ of Corti in a few minutes. So on the other point now to form this spiral organ of Corti is that initially the cells of the cochlear duct are similar, as we saw here. These cells are similar. These cells are similar. They haven't differentiated. But with time now, these cells, which are going to form the spiral organ of Corti, will start to differentiate. But before then, the first thing that will happen before these cells differentiate is that you'll have formation of this tectorial membrane. The one in blue, that is a tectorial membrane. So this tectorial membrane is one that covers the spiral organ of Corti proximally. Over time now, there is formation of two ridges, the inner ridge. This is the inner ridge, and then this is the outer ridge. So the inner ridge is a future spiral limbus, and then the outer ridge is now forms the inner and the outer hair cells. So this is the outer ridge. You have the spiral limbus, and then at this point actually you can start to see that there are some nerves that are being formed. And then these nerves now are the one that will come and form the cochlea now. So with time, you can appreciate the developments that will happen. This inner ridge will give us the spiral limbus. Then you have this sulcus, or the ridge coming across, spiral sulcus. Then you have now the outer ridge giving us the inner and outer hair cells. Remember, we usually have one inner hair cell and three outer hair cells. In addition to that, we'll also have some other supporting cells, cells of Hansen, cells of Claudius, pharyngeal cells, that is inner and outer pharyngeal cells, and then talk also about the pillar cells, inner and outer pillar cells. So they will develop at this point. So this is the ectoderm membrane, you have this spiral sulcus, spiral limbus, and then you have the inner and outer hair cells, and then you have even the supporting cells. You can also appreciate at this point now the neural cells will start to form the auditory nerve fibers to give us now the afferent sensations that come from the hair cells. And maybe to finalize is that the outer ridge is this one now, is covered by the tectorial membrane, giving us now a definitive organ of Corti. Now, at this point, I've alluded that impulses are transmitted to the spiral ganglion now, by this cochlear nerve to the central nervous system. I guess at this point that's obvious. And then now we're supposed to talk about how now the utricle forms its structures. Is that The utricle and the formation of this semicircular canals is that the utricle in the 6th week develops flattened out pocketings. So remember here the circle. So on the other side, you'll have the utricle. So the utricle develops flat out pocketings. And then this can figure out this is a utricle. Develops 3 flat out pocketings. These 3 flat out pocketings will undergo flattening. After flattening, what will happen is that they will oppose each other. They will oppose each other in an angle of 90 degrees, and then the inner point of it will disappear. So these are walls of the central portion of the outpocketing, opposing and then it disappearing. So we have a utricle here. It will develop 3 flat outpocketings. After that, these 3 flat outpocketings on the central portions of them is that you will have the central portions opposing and then disappearing to give us 3 semicircular canals at an angle of 90 degrees. So, but these semicircular canals are attached to one another, as you can see on this point. And then there are some thickenings on the distal aspect, or some of them. So how do those 3 thickenings appear? So on one end of the canals dilate. One end, for example, this superior semicircular canal, one end of it dilates, the other one doesn't dilate. When we talk about dilating, means widening. So one end of the canal dilates, that is the crus ampullare, while the other doesn't dilate. And that is now the non-ampullare side, the crus non-ampullare side. So this is a crus ampullare side, this is a crus non-ampullare side of this superior semicircular canal. On this other side, you can appreciate the same thickening happening here, and then the other thickening at this point. So this crus ampullare, now they form a crest. Inside it will have a crest, which is crista ampullaris. And then this crista ampullaris is one that now will be containing the hair cells for equilibrium. So cells on the walls of the circle and the utricle develop macula acousticae. So remember, you'll have the utricle here, and then you had the circle from our previous slide. So the macule, the circle, the utricle will develop into macula acousticae. And then now this macula acousticae and the crista ampullaris, they will develop the sensory hair cells that will be now in charge of equilibrium. So just a point to remind us is that we had utricle from the autocyst, circle from the autocyst, and then we've developed these semicircular canals. And then these semicircular canals, we have these dilated ends, which are crus ampullare. These crus ampullare, they develop a crest that is known as crista ampullaris. This crista ampullaris, together with the circle and the utricle, will develop some cells. These cells are now the sensory hair cells for equilibrium. And that's how now the sense of equilibrium comes across. Let's speak about the statoacoustic ganglion. For this statoacoustic ganglion is that we'll have these cells of the autocyst differentiating from the walls, as you can see here. So this is statoacoustic ganglion developing from the inner wall of the autocyst or the otic vesicle. So during formation of the otic vesicles, some cells will break from the wall to form statoacoustic ganglion. And then now this statoacoustic ganglion, the supporting cells will come from the neuronuclear cells. Just to say that this statoacoustic ganglion divided into 2 parts. It will divide into cochlea and the vestibular portions, of which now the vestibular portion will supply the utricle, circular, and the semicircular canal, which are in charge of equilibrium. That is the vestibular part. And then the cochlear part of the spiral part will now form the spiral ganglion or the cochlear ganglion, which now supplies now the organ of Otic. I hope that tries to simplify things. So we'll form a tympanic cavity. We're going now to the middle ear. The tympanic cavity is actually one of the simplest structures to be formed, given the fact that we have this is the first pharyngeal cleft. Then we have the pouch. So what will happen is that we have can appreciate this is a developing inner ear. Now we want to develop the middle ear, starting with the tympanic cavity. So this is a cleft. Cleft will form the outer ear, and then the pouch will form the middle ear. How? So we'll have now the first pharyngeal pouch, giving us this tubotympanic recess. So the pouch expands laterally on the outer part, and then distally it will expand outwards and distally to give us the tubotympanic recess. After having this tubotympanic recess, what will happen is that this tubotympanic recess will widen. It will widen, and you can see in this slide, the tubotympanic uh, recess, it will widen. It will widen to give us the primitive tympanic cavity. It's a primitive tympanic cavity. So this primitive tympanic cavity on the proximal end, it will remain narrow and connected to the eustachian tube. And that's the reason why this eustachian tube at this point will be called pharyngotympanic tube, because it will be connecting the tympanic cavity with the pharynx. So it will remain proximal, it will remain narrow, but now on the distal side, remember that is the side that is widening. And that you can appreciate well here. So you have the pharyngotympanic tube or the eustachian tube. It will widen to cover up this malleus, incus, and stapes, the ossicles. So what then happens so that you can have this? At this point, you can appreciate that this is a mesenchymal condensation to give us these ossicles. So these ossicles, it means that something will happen for them to be inside this middle ear. So what will happen for them to be the middle ear is that, okay, before then you have the tube, this tube allowing the communication between the nasopharynx and the tympanic cavity. So let's talk about this, the ossicles. So the malleus and incus from the first pharyngeal arc, stapes from the second pharyngeal arc. So that is now from the pharyngeal apparatus. We already know that the malleus and incus comes from the first pharyngeal arc, stapes comes from the second pharyngeal arc, and you can see them here. So this is the macular cartilage. You can see them, incus and the malleus, first pharyngeal arc, then the stapes, and also the hyoid bone are coming from there, and the stellate process coming from the second pharyngeal arc. But what is relevant for us, the ossicles, malleus, and incus, first pharyngeal arc, stapes from second pharyngeal arc. So they usually embedded in the mesenchyme till the 8th week, where now the primitive tympanic cavity will uncover them. So they usually embedded in the mesenchyme till 8th month, where the tissue dissolves. When now this tissue dissolves, it will form now the definitive tympanic cavity. So you can name these were covered in the mesenchyme. Then you had now widening of this primitive tympanic cavity to uncover them, giving us now, first of all, a permanent or this developed tympanic cavity covering or inside it the malleus and the incus. But before then, you can appreciate you have some ligaments and some muscles attaching to it. Where do they come from? So the endoderm of the primitive tympanic cavity extends over the formed spaces I had alluded earlier, but before then, the muscles formation for the tensor tympani, which is a muscle now for the malleus and incus, comes from the first arc, which is now being supplied by the mandibular branch of the trigeminal nerve. Then the stapedius muscle, which now attaches to the stapes. Remember, the stapes comes from the second pharyngeal arc. So the stapedius muscle will come from the second pharyngeal arc, and it will be supplied by the facial nerve. So at this point, also can give us a summary to go and check on what we talk about and what do we refer when we talk about hyperacusis. For the external ear, that's one of the simplest thing for us to form. So we have external auditory mediators. So you have the external auditory mediators. So these external auditory mediators comes from the first pharyngeal cleft, that is the ectoderm. So the middle ear comes from the endoderm. Outer ear comes from ectoderm. So what happens is that you'll have so what will happen is that you will have the cells at the bottom of the cleft proliferating. The cells at the bottom of the cleft proliferating to give us solid epithelial plate that is known as a meatal plug. And then this is a meatal plug. You can appreciate this is a developing middle ear. This is a developing outer ear. Developing middle is coming from the endoderm. Developing outer ear is coming from the ectoderm. So this is a first pharyngeal cleft. This is a first pharyngeal pouch. So the first pharyngeal cleft gives us the external ear, the external auditory meatus. So the cells at the bottom of this cleft will proliferate to give us a solid plug that is known as meatal plug. So this meatal plug will supposed to disappear in the seventh month, but if it doesn't disappear, what will happen is that we'll have congenital deafness. So imagine this meatal plug supposed to disappear, but it doesn't disappear. So it will give us congenital deafness. So the seventh month, the plug disappears and the epithelial lining of the floor forms us, gives us the definitive eardrum, or this external auditory meatus. Remember, the eardrum is also known as the tympanic membrane. So the tympanic membrane will be formed at this point. So for the eardrum or the tympanic membrane, now it's being formed by portions of the cleft and portions of the pouch. So that means it will come from the three uh, it will come from the three germ layers. The ectodermal epithelial origin from the outer part now, and the inner part, that is the endodermal epithelial origin of the tympanic cavity, and then the middle part, which is actually a very thin part, the mesodermal intermediate layer, gives us the fibrous stratum. So that the eardrum comes from ectoderm, mesoderm, and endoderm. Ectoderm where? Outer part. Mesodermal part, the middle part, the fibrous stratum, and then the endoderm is actually now the epithelial origin of the tympanic cavity. So the membrane separates the external auditory meatus from the tympanic cavity, and what function does it play? Now is on that does the work of amplifying, as you can see here. So this is it. Okay, amplifying is done by this malleus, incus, and the stapes. For it, it's the one that just enables, first of all, separation of the outer ear to the middle ear, and then it also prevents some other structures from getting into the ear. Yeah. So you can see here we have the three germ layers, the ectoderm, mesoderm, and endoderm, giving us the tympanic membrane. And then maybe I can give us an assignment to go and check on this. Go and check on how the eardrum presents, be able to draw the parts of the eardrum. You can be asking an exam setting. Maybe to maybe to maybe to try and finalize is that we'll have the auricle. This auricle will develop from the first pharyngeal arc and the for the auricle, it will develop from the first and second pharyngeal arches of the cleft. That means it comes from the neural crest cells. So the auricle is the outer part. So how will it develop? So you'll have these neural crest cells on these two pharyngeal arches proliferating to give us six auricular hillocks. After that, now the second arc now makes almost the entire external ear, except the trigger, which gives us the first pharyngeal arc. So whatever happens that will have duplication, pits, and tags occur due to gene misexpression and mutations. And you can see here, you have the six auricular hillocks coming from the first and second pharyngeal arches. The second pharyngeal arc proliferates, and then remember before that you'll have tissue proliferation to give us six auricular hillocks, but before then, the second arc proliferates more to give us almost the entire external ear, except the triggers. You can have duplication, pits, and tags due to misexpression. And that is what now we have the anatomy of the outer ear or the auricle, as we talked about the lobule, the triggers, close of the helix. You have the close of the antihelix, and then you have the helix there, as you can see there. Maybe to finalize up is that you have some anomalies at this point. So we'll have some congenital anomalies, starting off with this microtia. So microtia means smaller ears, of which you have various degrees: first degree, second degree, third degree, and fourth degree. Fourth degree now is being called anotia, absence of the outer ear, or recalled completely. At this point, you can appreciate the tags being present. There's a pre-auricular sinus and there's a aural atresia. Other anomalies that you can appreciate is that you have cretonia, constricted ear, Star's ear, hemangioma ear tags, and cauliflower ear. So maybe another point to appreciate is that the two primary anomalies of the ear is actually the sensory neural and conductive deafness. So maybe at one point you can go and check what we talk about. We talk about sensory neural and conductive deafness. What do we talk about? So maybe that's an assignment that I can give us at our own time. We go and check. We talk about that. And maybe finally is that the ear is the avenue to the heart. So find it at a point where you know what you're hearing and what is you're directing to your heart. So with that, we can call it a day. Yes, have a wonderful